Hey everyone, Dr. Marie McNeely here, welcoming you to episode 867 of People Behind the Science. Today I am joined by our guest, Dr. Sydney Glassman, and we are going to have a fantastic time chatting about life and science. If you like this episode and want to check out more stories from today's scientists, you can subscribe to our podcast on your favorite podcast platform, and I would be delighted if you have a moment to leave us a rating or review to let us know what you think of the show. So without further delay, let's get our conversation started with Sydney, and she has some exciting experiences to talk about today. So listeners, get ready to meet another one of our amazing people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Sydney Glassman. So, Sydney, welcome to the show today. How are you?
Hi, I'm good. Thanks for having me.
Well, thanks so much for joining us. We're excited to learn more about you and the work that you do during our conversation today. But I'd like to start by giving our listeners a little bit more information about your current position and the steps that you took to get there. So listeners, Sydney is Associate Professor in the Department of Microbiology and Plant Pathology at the University of California, Riverside. She completed her bachelor's degree in biology with a concentration in ecology and evolutionary biology at the University of Pennsylvania. Next, Sydney received a master's of environmental studies degree in environmental biology from University of Pennsylvania, and she was awarded her PhD in environmental science, policy, and management from the University of California, Berkeley. Sydney conducted postdoctoral research at UC Irvine before joining the faculty there at the University of California, Riverside in 2018. And Sydney, we're excited to get to know you as a scientist, certainly in our conversation today, but also more generally as a person. So can you start by telling us what you like to do when you're not doing science?
Well, right now, I spend most of my time with my 1- and 3-year-old and my 2 dogs.
Aw.
So that's pretty much all of my time right now is my children and my dogs.
I love it.
And my husband.
You can't forget the spouse as well. So what are your kids' favorite activities these days?
They like to read books. They're really into reading.
Awesome. And then you mentioned dogs as well. What kind of dogs do you have?
They're both mutts. One is 115 pounds and one is 15 pounds. So they're a little bit of an odd couple. I got like an extra small and an extra large. They're both mutts. One is like every big dog mix you can imagine. And one is like every little dog mix you can imagine.
I think that's amazing. Well, it's wonderful to hear a little bit more about your life outside of science. And it sounds like you keep quite busy managing 2 young children, plus the dogs, plus spending some quality time with your husband, which I think is important to take you out of the lab so you can kind of get a break from the science. But we'd love to talk about your work next. So, Sydney, how do you describe your research to someone who's not in science or not familiar with your field?
So mostly what I study is the impact of wildfires on soil bacteria and fungi. I'm really curious about how fires impact soil microbial communities and how those things help ecosystems recover after disturbances. And I'm interested in fungi and specifically mycorrhizal fungi. So those are symbiotically associated with plant roots. So I like beneficial relationships in nature, fungi that are symbiotically or positively associated with plant roots. Those are mycorrhizal fungi. And also I study different types of bacteria and fungi, and I find fungi really fascinating.
Oh, I think that's awesome. And I know the work that you're doing looking at recovery after wildfires is particularly important there in California, where this is a big issue. So I look forward to talking more about your work as we go through our conversation today. But I'd love to take a pause to talk a little bit about motivation. I think in science, there are so many challenges that arise that it can be helpful to have a little quote or a saying or a mantra, just something that keeps you motivated and inspired when times are difficult. So Sydney, do you have a favorite quote or a saying or something that you can think of that really keeps you motivated?
I don't know if I have a mantra exactly. My PhD advisor used to say mycology without apology. I don't know that I think of that as to help me keep going when things are difficult, but I always think about how fungi is sort of like an underdog in biology, and I'm here to understand them and bring light to them and get people to understand why they're so cool and interesting and fun and why there's so much important stuff to know about them. But I think rather than a mantra, I just think about there's just so many reasons why I got into what I do. And I think about all those things. And how fungi are really important for many aspects of people's everyday lives and for society. And people don't know that, but they are. And I like to study them to bring light to that and hopefully solve some problems. And then I also think a lot of what I do is important in terms of like teaching and mentorship. And that also keeps me going.
Definitely. So can you talk a little bit about this love of teaching and mentorship and maybe even outreach as well, if you do any, to spread the word about how awesome fungi are?
So I created from scratch a course at UC Riverside called Field Mycology. I take students out into the woods and teach them how to mushroom hunt. Most of these students have never been hiking before. They definitely have never been looking for mushrooms before. So I get them excited about mushrooms and we take them outside of Mushroom ID. We do a lab where we learn how to do mushroom identification. And then in my lectures, we teach them all about fungal diversity, all about fungal ecology, and all about fungal impacts on society. So like today, my lecture is about fungal hallucinogens. Last week, I was talking about fungal poisons and fungal medicines and how fungi do have influences on human medicine, on poisons that affect humans. And also today is about hallucinogens. And then I also talked about baking and brewing last week as well.
Well, sounds like an amazing course. Well, Sydney, thank you for sharing a little bit more about the teaching side. What about mentorship? Can you talk a little bit about how you work with students?
Yeah, so I have several PhD students. I've had 6 PhD students so far. 3 have graduated. I have 3 currently in my lab. And I've had a ton of undergraduates come through my lab, over 20. I have a lab manager. I've had multiple technicians work for me and multiple postdocs as well. So I basically meet with everyone one-on-one every week in my lab, make sure that they know what they're doing. And I don't know, mentorship spans a lot of different things. It goes from everything from teaching them the basics of fungi and the basics of microbiome science or statistics or helping them with the writing to working on their confidence. And then also once people leave my lab, I usually still mentor them forever. I mean, I still keep in contact with all my graduated PhD students regularly. I help them. I write letters of recommendation for them. I advise them on various things. We still work on papers together after they leave as well. So mentorship takes up a lot of my time. And then, yeah, I do have a lot of undergrads who've come through my lab as well. Some of them have won fellowships. 2 of them won these like really prestigious UC-wide fellowships to do research on these in our natural reserve system. So that's this system that not a lot of people know about, but there's about 40 natural reserves that are owned by the UC system all over California. And this was a fellowship to help them do research fieldwork in one of them. So I had 2 students who won these fellowships to do fieldwork in these natural reserves. And as I said, it does take up a lot of my time and is something that's important to me to mentor the next generation of mycologists.
Oh, that's so cool. Well, it sounds like you've been a role model and a mentor for so many aspiring and early career scientists, but I'd love to talk about some of your role models and mentors as well. So do you have particular people who you look back and you say, wow, those people really had a big impact on me and on my career as well?
For sure. My master's advisor, my PhD advisor, and my postdoc advisor definitely had the biggest direct influence on me. I probably wouldn't have gone as far as I have in my career if I didn't have so many wonderful mentors. I currently have mentors too at UC Riverside as a faculty, but it's really important to have an academic family of people who are looking out for you. But my PhD advisor is really the person who made me a mycologist. I already mentioned how he says mycology without apology, but he's the one who taught me how to identify mushrooms, who taught me everything I know basically about fungi. He taught me so many things. He made me a better writer, that's for sure. We went on the field together so many times. Like there were a lot of times where once we were hiking in Yosemite and we got lost, we were just like totally off trail. There was snow on the ground. We were like hiking through snow. We had to like climb up this mountain and like we had to scale these rocks.
Oh no.
And I was like, wow, if my like 60-plus-year-old PhD advisor can do this, like I could do I better not wimp out on this. I was like in my 20s and he was like definitely at least 65, literally scaling up rocks. And then I was also thinking, okay, he's not going to let me die. Eventually we found the trail and then we saw that the trail had been closed due to snow. We went on a lot of wild adventures. He and I traveled a lot. We did a lot of field work together in a lot of different places. And he definitely had a huge influence on me. My postdoc advisor also, she had a lot of influence on me too, especially in the way that I run my lab. I take a lot of influence from her in terms of like how to be a mentor. Mentorship is really important to her. And I definitely take a lot of steps from her playbook in terms of how I run my lab and how I mentor my people.
Well, that makes sense. And Sydney, it sounds like you've had some fantastic mentors and advisors throughout your career. And I'd love to talk about where your story begins. Can you share what first got you interested in science and maybe got you started on this path?
I I would say probably in high school, I really loved my AP biology course. My high school AP bio teacher probably was the biggest starting point for me in terms of getting into biology. Then I also did a semester away program. When I was in high school, I lived on a farm in Vermont. I did this thing called the Mountain School, where I lived on a farm in Vermont for a semester of my junior year and spent a lot of time thinking about where our food comes from and learning about how to read the forested landscape. I spent a lot of time in the woods. I'm from LA. Like, I really didn't have that much of a background hanging out in the woods. I had a very urban environment growing up, but then I spent a lot of time in the woods thinking about how important it is, thinking about how to read the forested landscape. And that sort of set me on this path to want to be either an environmental science or a biology major when I went to Penn. And then when I went to Penn, I realized pretty quickly that I wanted to be a biology major and I wanted to do science. My freshman year, I spent part-time doing research in Brenda Casper's lab and part-time working for the EPA. And I realized I really liked doing research. So working with Brenda Casper really also was like a pretty formative experience for me. I worked on arbuscular mycorrhizal fungi, which are symbiotically associated with plants. And that really got me really appreciating and understanding positive relationships in nature and how they have such an important influence on our environment. 80-90% of plants are associated with mycorrhizal fungi. They're absolutely critical for plant survivorship. They actually, evolutionarily speaking, help plants colonize land. So they're really important. And this is really a beneficial relationship that's super widespread. It's the most ubiquitous terrestrial symbiosis. So that really set me on my path of really wanting to get into fungi. And then I studied abroad my junior year of college in Costa Rica. And that's what got me really into fungi. So I was hiking around in the woods and I started to notice all the mushrooms. I remember we were like out on this night hike trying to look for, we were out herping. So that means we were looking for reptiles and amphibians because they're more active at night. And I kept asking my professor about, oh, what's this plant? What's this mushroom? And she's like, Sydney, we can look at those during the day. Look for hers. And I really realized that fungi was really my passion. Like, I just started to see how beautiful they were. There were mushrooms everywhere.
Sure.
I studied mycorrhizal fungi for my thesis there. So that's when I got really into fungi. Basically, being in Costa Rica was what made me want to be an ecologist. At first, I wanted to be a tropical field ecologist. And then I decided for my PhD, I really wanted to join a mycology lab and become a mycologist. So then that sent me off to go to Berkeley and do my PhD with Tom Bruns, who's a mycologist. And well, it wasn't really practical to do research in the tropics at that point. So I did local research, but that was my path of how I got into fungi.
Oh, interesting. And your PhD was in a program that was environmental science policy and management. Can you talk about how having this policy and management framing also maybe has shaped your research?
I can't remember if it's SNL or Monty Python where the title was, it's neither this nor that. They give a name and they're like, oh, it's not this, it's not that, or it's anything discussed. That's kind of SBUM. Literally no one in SBUM does environmental science policy or management. It's like the worst name ever. It's a complete misnomer for the department, I think. So I did fungal ecology. My PhD advisor was in this plant microbiology department and I was a fungal ecologist. And like most of the people in this department, I don't think even anyone does policy or management in that department. So I don't know.
It's a vestige of the past, perhaps. Yeah.
It's just like a name that I don't think really explains anyone in that department. And it's a huge department, but a third of the people are doing environmental science or like ecosystem science. And then a third are doing organismal biology. And then a third are doing society and the environment. But yeah, I don't really think that name encompasses it.
That's fair. And then you decided to do postdoctoral research there at UC Irvine. Can you talk about your experience there as well?
I really, really loved being a postdoc at UC Irvine. It was such a different experience. I went into a lab where they were primarily working on bacteria. So I learned a lot about bacteria at that point, and that actually really set me up for my current lab at UC Riverside, where I pretty much work on fungi and bacteria kind of equivalently. That was where I really learned about bacteria and started getting into bacterial culturing and bacterial sequencing and learned that pretty much all the methods we use for fungi, you can apply to bacteria as well. So at that point, I brought bacteria into my research when I started my own lab. And then it was a really good experience for me to see a different lab, how a different lab functioned, to see a different mentor and how they function, because everyone sort of has their own research styles. And so I think it's good to be shaped by multiple mentors and see different ways that different labs function to get an idea of what you want to do in your own lab. Just like even the way the labs were set up were pretty different. In my PhD advisor's lab, there's a bench for DNA extractions and there's a bench for culturing. Every bench had its own task. Whereas in my postdoc lab, everyone had their own bench to do their own work. So that was a pretty different approach to setting up your lab. But I don't know. I really loved being at Irvine. I loved the people there were really awesome. I developed a lot of really good relationships, both with my own advisor, Jen Martiny, and then also some of the people we worked with, like Steve Allison. I still collaborate with him now. I made really good friends. I also just really loved living in Irvine. It's just a really good experience for me. And unfortunately, it was too short. Basically, 6 months into my postdoc job, I got an interview to do a faculty job. So I actually got my job offer for UC Riverside. I was only 8 or 9 months into my postdoc. So I actually had to negotiate to stay at Irvine as long as I did. So I was only there a year and 10 months. And honestly, I would have loved to stay longer because I really did love living in Irvine. I really did love working with Jen Martiny. I liked the research we were doing. And yeah, I was really happy there in general. So I would have stayed longer if I could have. But yeah, I was only there a year and 10 months.
Oh, I think that's awesome. It sounds like the faculty job search went well. And I think this is often a sticking point in scientific careers. So can you talk a little bit about what that process was like and perhaps why you started it so early into that postdoc?
I guess I'm just a very proactive person and I'm just the epitome of a go-getter type A person. If there's something to apply to, I'll apply to it. And I always figure there's no harm in trying. And I figured the sooner I started applying, the sooner I would get experience. And then I figured the more applications I filled out, the more feedback I could get, the better my applications would get each time. If I got the chance to do interviews, that I would get improved during them. And I just figured you never know when these jobs are going to come up. So if I felt like if there was a job that I was a fit for, I would apply to it because it could be that that job would never come up again for 20 more years. And then also, what did I have to lose? I mean, the worst that could happen is I don't get an interview or I get rejected. And I knew I had a pretty competitive background. I had published 4 first author papers for my PhD, and I had a bunch of collaborative papers as well. And then I had a ton of data to do more papers. And so, and I had published a first author paper for my master's as well. And I also had a pretty good track record of funding. I'd funded myself with this NSF GRFP, this really competitive fellowship during my PhD. I'd won another grant, this NSF DIG. That was another competitive grant. I won a lot of awards during my PhD. So I figured I had at least some shot because I had this good track record of funding and papers, which is what you need. And then I also had a lot of experience teaching. I had a lot of experience doing mentorship as well. I had a ton of trainees. When I was a grad student, I had at least 9 undergrad mentees. And I did a ton of outreach too. So I figured I had all the things that you needed to get a job. And then I might as well just start applying. And since there were a bunch of jobs that were fungal specific, I was like, what do I have to lose? I'll just apply. And I applied all over. I wasn't thinking I'm going to be specific in where I'm applying to. I applied to jobs. Anywhere there was a fungal job, I applied. And that was all across the country. And then I was so shocked when I got interviews. I was even more shocked that when the interviews I got were at UCLA and UC Riverside because I was from Southern California. And I really never imagined that I would be able to get a faculty job close to where I lived because I always thought, okay, it's really hard to get a faculty job. And then it's even harder to get a faculty job close to where you're from. My first interview was UCLA, and that's literally 10 minutes from where I grew up. I was so shocked at the time. I couldn't believe that I got these interviews. And then I got the UCR job.
That's awesome. So can you tell us a little bit more about your lab at UCR and perhaps if there's a particular project you're working on at the moment, Sydney, that you were just the most excited about?
So my lab at UC Riverside, I just came out of a period where I had some really large grants where I was working on a bunch of big projects. And now I'm sort of coming out of that, did a ton of research, and now I'm gathering all the results and trying to understand them and publish them and write them up. One of the projects I was working on was I had a large $3 million grant from the Department of Energy, and that had a lot of different aspects that I thought were really cool, and there's a lot of room for follow-up. So one aspect is I have been developing this culture collection of pyrophilous bacteria and fungi. So these are things that survive fires. And what I did was I collected soils and smoke and mushrooms that appeared after fires, and I isolated bacteria and fungi from them. So I developed this really large culture collection of bacteria and fungi, and I really wanted to assay their traits, figure out okay, what is it about these things that enable them to survive fires? So I did a really large project sequencing their genomes, their transcriptomes, and doing some biophysical assays on them, trying to figure out what are their traits. So that has led to some really interesting results, figuring out that at least for the fungi, some of them are really good at growing fast and others are really good at eating charcoal. And there seems to be a trade-off such that the ones that grow fast are less good at eating charcoal and the ones that eat charcoal don't grow as fast. And that was really cool to see that they definitely do have the genomic machinery to eat charcoal. Another cool part of that project is that wildfires are hard to study. They're really unpredictable. It's really hard to get good replication with them because you don't know when they're going to burn. And then when they do burn, they're different sizes, they're different severities. So what we've been doing in my lab is we've been setting fires in buckets. We call them pyrocosms. And we take soil, we put it in a bucket, and then we manipulate the amount of charcoal to make a hot or a high intensity or low intensity fire. So we make a hotter fire by putting more charcoal, and then a less hot fire by putting less charcoal. And then we've been taking soils from different environments. So we took soils from all these different environments that had different natural fire regimes. So we took chaparral shrublands, which normally have like pretty high intensity, like infrequent fires. We took fires from grasslands, which have usually pretty low intensity frequent fires. And we took soils from coniferous forests, which also have pretty infrequent high intensity fires. And then we took soils from all these different environments, and then we put them in pyrocosms and we set high and low intensity fires. So we put the same fire parameters for all of them, and we set the fires, and then we were looking at what happens to the soil microbiomes, what happens to the bacteria, the fungi. We also collaborated with someone at UC Davis to see what happens to the viruses. So we're looking at bacteria, fungi, and viruses after these fires of different intensities, and then also looking at nitrogen cycling at these as well. So we've been setting a lot of fires in buckets and trying to study how the bacteria and fungi and viruses and greenhouse gas emissions change after these fires. So that's been a fun project.
Well, Sydney, that sounds awesome. And I love the word pyrocosm. Did you coin that?
Actually, that's my PhD advisor. He is the one that came up with that.
Okay, that's awesome. And I have to ask, where are you conducting these experiments? Are these being done in the lab, or is this like somewhere in the parking lot where you've got all these buckets full of fire?
UC Riverside has an agricultural experimental station. I don't know how many people are aware of the hatch system and the land grant system. So in the 1800s, there was a law that instituted this hatch program and these land grant institutions through the USDA. So every state has at least one, and California has several. We have UC Riverside, UC Berkeley, UC Davis, that are all part of this land grant institution. And as part of that, we have this agricultural experimental station where we do research that specifically is aimed at helping with agriculture and natural resource management. Right next to campus, we have this really large agricultural experimental station. There's tons of citrus trees where people are doing research on citrus. There's so many different plants, but there's also just these big flat areas that I basically found like a big platform where it was like a place that you could not accidentally set a fire. And I'm setting fires there.
That's awesome. Well, this sounds like a really cool project. So you mentioned that you're perhaps wrapping up with data collection and starting to get the results together. Do you have any ideas of what the results are going to tell you?
So we published a paper in PNAS in January for the pyrophilus fungi. We were looking at where I mentioned about how some of these fungi have the genomic machinery to eat charcoal. And the ones that can eat charcoal do so at the expense of growing fast. We're also working on publishing the results on the bacterial genomes. And we showed there that these bacterial isolates that we isolated from fires are actually really distinctive from even basically you can have the same species of bacteria, but one that was isolated from a burned environment versus one that wasn't, like has a lot more machinery to be able to degrade charcoal. So they're just like really plastic, these bacteria, and are capable of taking on different genetic capacities through things like plasmids. And then in terms of the pyrocosms, we've been seeing that my favorite result so far is that there's sort of this famous adage in microbial ecology, this concept of everything is everywhere, but the environment selects. This was back in the 1800s. This guy named Bosbecking came up with this idea. He said, there's no dispersal limitation in microbes. Every microbe is going to be everywhere. And every microbe is able to just produce so many spores that can get anywhere they want. And it's just that the environment selects. And then we sort of debunked this very thoroughly during my PhD period. We realized there is a lot of dispersal limitation, like different microbes in different locations are different. If you're in California, you're going to have different microbes than in the East Coast because there is distance between them and the microbes can't just move across the country that easily. But in terms of pyrophilus microbes, actually, I think there's just like so many years between fires that these things can build up over time. So you have these fire resistant or thermotolerant spores or these things called sclerotia, which are these kind of bits of hyphae that are really resistant to a lot of things and can just like sit around for years. So I think that even if the dispersal events are rare, they can sit through time so they can get everywhere and wait around for a fire to sort of select them. So once you have a hot enough fire, they do actually grow because everything else dies. And then these things survive and they are able to sort of take over completely and totally dominate the environment. So what we found is that after a grassland where you wouldn't normally see pyrophilus microbes dominating like a normal grassland condition, the pyrophilus microbes go crazy. Like they're like 90% of the community. And same thing with the chaparral. Like there's tons of these pyrophilus microbes and you kind of get the same microbes in both of these locations, even though they're really different biomes and different locations. So that was something that I found really cool.
Interesting. And I think, Sydney, from your description, it sounds like everything went smoothly. You did these experiments, you got some really cool results. But I think that's not always how things work in science. I think people don't often get to see the struggles, the troubleshooting, the failures that happen. So we love sharing these stories because I think it is so important for people to have an accurate picture in their mind of what the process of science is like. So do you have an example of a big challenge that you faced or a major failure that you had? And if so, could you walk us through that experience?
I mean, how much time do you have? First of all, we had to do so much troubleshooting for these pyrocosms. We spent a whole year just doing troubleshooting on them and just trying to figure out how to get them have reliable temperature profiles. And we did a lot of troubleshooting because we're like, oh, first, should we bury them or should we not bury them? Does it matter if they're open to the air or in the soil? Like, does that change the temperature profiles? And then we said, okay, how much charcoal do you have to put? And then when you put it in different soils, like, is it actually reliable? Well, it turns out that across Fastlands and Chaparral, like the soil's pretty similar. And we did get pretty similar temperature profiles. But then when we put it in coniferous forest soil, the soil burns so hot. They have so much carbon in them that the soil burns so much hotter with the same amount of charcoal that we had put for our high intensity fires for the Chaparral and Grassland. So that took a ton of troubleshooting. We had to do a ton of more burns to figure out how to get the same temperature profiles in those soils because they just burned a lot hotter. But there were a lot of things. I mean, you have to get the right weather because you can't do these when it's raining and you need to have the smoke quality be fine. And we had to get permission from the county such that it wasn't too hot, such that it wasn't too windy, such that it wasn't too bad air quality already. We had COVID during this time, of course, like that shut down my lab for like a full year. I had 2 babies during that time as well. So that took me out for a summer, but luckily I had good employees still working. There were a lot of things that happened, both in my own personal life where I had 2 kids and then in COVID, but then also we had to do troubleshooting to make sure that these things worked. And then, of course, getting the DNA to come out of these soils wasn't that easy either because you really burn them and everything dies. And so just even getting any DNA to come out was hard, especially with getting RNA out. That was still an ongoing challenge. Getting the RNA to survive these fires has been really, really challenging to look at gene expression. So there's been like a lot of different aspects that have been challenging.
So what kept you going and kept you from just saying this project isn't going to work?
I don't know. I just think that if you have that attitude, you're not going to succeed in science. Like, you just have to learn how to be persistent and to make lemonade out of lemons. Like, I think that that's something that I've always been really good at, is making lemonade out of lemons. Like, I didn't start my PhD wanting to study fire. I was studying ectomycorrhizal fungi and their natural ecology, like what fungi are where and why. And then not once, but twice during my PhD, my plots burned down in catastrophic megafires.
Oh, no.
Then I studied what happens when your plots burn down in catastrophic megafires because I had pre and post fire samplings from that. And then in Riverside, when I started here, there was a big fire that happened like right when I started my job. And basically every year there's all these fires. And so I was like, okay, fire is a really important thing. I should study that. And then I've been studying these plots for 6 years after the Holy Fire, doing all this long-term monitoring on them. And then my plots burned in the 2024 airport fire. So it wasn't just that my plots kept burning, my plots started to burn again. So then now I'm pivoting and I'm studying reburns. I'm studying, okay, what happens when your plots reburn? What happens when things burn again before they would naturally be able to recover? What's that? So that's like what I'm studying now. So I feel like whatever happens, I'm always going to pivot and be able to turn it into something.
Well, Sydney, I absolutely love that. And I think you're right. You have to get creative with these solutions and you have to be able to adapt to the changing environment of your scientific research program, but also just the scientific landscape more broadly, which I think it sounds like you've done a tremendous job of over the years. And we don't just want to talk about the challenges and the tough times. I'd love to talk about your successes as well. We've kind of hinted at a few of them throughout our conversation, but do you have a favorite success story that you'd like to share with us?
I can have a most recent success story. So just recently in the last couple of weeks, I was alerted that I was awarded the Alexopoulos Award from the Mycological Society of America. And that is an award for the most promising young mycologist. And that was really exciting for me to win that award. You have to be within 10 years of your PhD. And I just am at the cutoff because this May is going to be 10 years post PhD for me. And that was really exciting because the Mycological Society of America has always been my favorite society. And it's been something I've greatly admired. My PhD advisor had won this award 40 years ago. And it was always something that I really aspired to be recognized by the society. And it does feel really good to be recognized for my contributions in the world of mycology from the Mycological Society of America, which is the society that I've kind of grown up with.
I think that's awesome. So did this come as a surprise or did you kind of know that you had maybe been nominated and had a chance at it?
I knew that I was nominated, but I knew I'd been nominated multiple times and hadn't gotten it before. So I never knew what was going to happen. I'd been hopeful, but yeah, I had been nominated multiple times and hadn't won it. And that's sort of something that you have to get used to in science is just rejection. You kind of just have to develop a really thick skin. Your papers get rejected, your grants get rejected. Students don't want to join your lab. They reject you. When you teach, you get bad evaluations. And it's just sort of something that happens and it sort of just goes with the territory. So I knew I had been rejected from this multiple times and hadn't won it before. So I wasn't 100% surprised, but it was definitely exciting.
Oh, absolutely. And is this one of the kinds of awards where they present it to you at the conference?
Yeah. So I'm going to go in July to the Mycological Society of America annual meeting and accept my award.
Oh, awesome. Where's the meeting this year?
Portland, Oregon.
Excellent. Well, we hope you enjoyed the celebrations and kind of basking in the moment of this big victory. And I think that can be tremendously meaningful, taking a step back, particularly in these relatively early career stages, to get that recognition of your contributions. And it can bolster you to continue on this path of success going forward.
It's definitely very exciting to win this. And definitely I need some bolstering after this period where I had a lot of money and now all my money has pretty much run out and there's not really a lot of grants to apply to right now. So I got to figure out how to fund my lab. That's always the biggest job of a faculty member is how to fund their lab. And that's definitely something I'm working on now. So it is nice to have that recognition and hope that I will get funding again.
Absolutely. I think it can be validating. It sounds like it might be time for a pivot again, which I feel like those are good ways to kind of reinvigorate your grants as well. And we talked about just some of the great work that you're doing. And I think these periods where you're in this transition and trying to apply for all of the grant opportunities that can come up can be very busy. And we try to encourage our listeners out there to take breaks and try not to let your work completely overtake your life. And one of the things that I think really helps is blocking off time to read for fun. So we love getting book recommendations from everybody that we have on the show. So Sydney, do you have a favorite book or one that you've read recently that you can recommend, whether it's related to science or not?
I really like fantasy. That's probably my favorite genre that I read outside of work. And right now I'm reading the Mistborn series by Brandon Sanderson. So I've been reading Mistborn and that's been an enjoyable book. It's been good for me to get back into reading. I had a period, especially with having 2 kids in the last 4 years where I wasn't reading very much, but I've been proud of myself that I've gone back into reading for fun in the last recent months. I've managed to do that. I sort of have my kid in bed with me and we read her books. And then once her books are done, then mommy gets to read her book and then she kind of looks for letters. That's like her new thing that she's been doing is looking for letters that she knows in my book. And then eventually she gets bored and falls asleep. That's been my new way that I found time to do some reading, actually.
I absolutely love that. And it's wonderful that you're reading with your kids every night. I think that's a good way to kind of get them excited about reading as well.
Oh, yeah. They really, really love reading.
I think that's phenomenal. And we talked about different aspects of your life as a scientist. And one of the things that you mentioned is you've been able to travel quite a bit to do field work. And I think this can be a really fun experience and also just going to conferences and presenting your work and meeting scientists from all over the world. And I think these travel opportunities are something that people don't necessarily associate with scientific careers. So do you have a favorite place that your science has taken you, Sydney, that you can tell us about?
Honestly, I traveled a lot in my PhD. I would say I've traveled less with faculty just because of COVID and then also because of having children. But during my PhD, God, I traveled so much. I gave a talk at the International Mycological Congress in Thailand, and that was really fun. I spent a whole month afterward traveling. I was in Bangkok for the conference, and then I went afterward and traveled throughout a bunch of different places in Thailand. I was in the jungle there. I went to Laos, and I spent time in the jungle there. I went to Vietnam, Cambodia, Singapore, and Malaysia during that time. So that was a really, really cool trip that I was able to travel all over Southeast Asia because of my research. I gave a talk at another conference that was in Prague, which was really fun. Oh, and I also went to Israel with my PhD advisor because we were also part of this grant where we're helping them work on fires and studying how fall versus spring prescribed burns affect soil fungi. And so we were helping them learn how to do the high throughput molecular sequencing that we use to study fungi. And that was also a really cool experience.
Well, that sounds amazing. In Costa Rica, we also can't forget that experience as well.
Oh, yes. That was during my undergraduate. I studied abroad in Costa Rica.
Amazing. Well, Sydney, it sounds like you've had some great travel opportunities. If our listeners are able to make it to Thailand, visit Bangkok or other places in Southeast Asia, do you have recommendations of things that just absolutely must make the itinerary?
I think if you're into the outdoors, going to the Khao Sui National Park, I don't know, I'd have to look up exactly what it was called, but there was this national park in the south where I got to see a slow-moving loris and a sun bear, and that was really, really cool. I really, really enjoyed that. And Laos was really cool too. I spent this weekend hanging out with this indigenous community, and being in the jungle there. And one thing I really enjoyed was talking to them about all the mushrooms. They just knew everything about the mushrooms. They're like, oh, this one you eat when you're sick, and this one you eat when you have a stomach ache. They just knew so much about all the different plants and the fungi and had a lot of knowledge about them. And that was really, really cool to see that. And also they had just like so many mushrooms in the markets. That was really cool. Like they really eat a lot of mushrooms there. So that was really fun for me to see all of that knowledge about fungi and the different things that they do about them. How many general listeners would want to do stuff like that? I don't know.
I'm interested. I feel like you'll just be disappointed though when you get back to your local grocery store and there's like 4 mushrooms. But this sounds like a phenomenal experience. And it sounds like you've had some opportunities to meet and work with local communities and people all over the world. And I think when you meet people in science, they often don't measure up to the stereotype that I think a lot of people have. This idea of a stodgy old person working in a lab all by themselves, this picture that you often see in movies and books and things like that. So we try to break some of the stereotypes out there by talking about this human side of science, whether it's quirky traditions or just funny or fond memories that you shared with colleagues. So Sydney, do you have a story of your own, whether it's about a lab tradition or just an experience that you shared with colleagues that you think goes against some of the stereotypes that you can share?
I think everything about our lives is not very stereotypical because we definitely aren't alone working in our labs. All of us have a bunch of people in our labs doing science together. Science is very collaborative. But I would say in my PhD, we used to do an annual beach trip where we did the fungal labs beach trip. So a bunch of different labs who studied fungi. We would go to Point Reyes and do like a big bonfire on the beach there and a picnic. So we did that every single year. And then when I moved to Riverside, I also tried to get another mycologist here to do that with me, where we would go to San Clemente Beach and bring food and do a picnic and go to the beach and basically do a fungal labs beach party.
I think that's amazing. I think it sort of necessarily takes you away from the fungi. So you have to talk to each other about other things because there's no fungi on the beach, right?
Well, you'd be surprised. There's fungi everywhere.
Really?
But you're not going to find like a lot of mushrooms on the beach. That's for sure.
What kind of fungi do you find?
Well, there actually are fungi associated with ocean water.
Okay.
And like with kelp too. But that's not my area of expertise.
Oh, that makes sense. Well, this sounds like a fantastic tradition and a good way to get people out of the lab and into the sunshine and taking a break for a little while. And I think that can be really important for recharging the lab and helping to build these bonds between lab members that I think can help solve some of these difficult problems that you're trying to solve in the lab. So if we took away the barriers that normally hold you back and you had unlimited resources, unlimited funding, time, staff, whatever you needed, Sydney, is there a particular question that you would most want to solve with these unlimited resources?
Certainly in terms of what I do now, just being able to fund myself unlimited to be able to understand pyrophilus fungi and their traits and how they're surviving is definitely my biggest area of interest in terms of current research. But probably if I could answer any question, I guess, how can fungi be used to solve environmental problems or like recover burned landscapes or take over polluted landscapes, or really that's sort of like an applied area I've been really interested in, is how can we use fungi to, or soil microbes in general, to restore environments and ecological communities.
Very interesting. And so for this question, is the main barrier a feasibility and logistics problem?
Yeah, there's logistics, there's feasibility, there's funding, are fungi the problem, or is it really more of like a social, economic issue? It's not just biology. Once you learn the science, you always have to have an application, and that has to do with economics. Can things be scaled, or are things feasible to people? Is there a will to do something like that? And there's a whole lot of other features and factors at play when it comes to working in lands that have a bunch of different owners, and you don't really know what all the stakeholders are there.
Well, that makes sense. Well, Sydney, I love this question, and I appreciate you dreaming big with us and giving us something to think about today. And I know we've talked about a variety of different things over the course of our conversation today, but we'd love to end by talking about advice. So is there a piece of advice that somebody gave you at some point in your career that really helped that you can pass on to our listeners today?
The most important things that really helped me be successful in my career is, as I was saying, making lemonade out of lemons. There's this concept of like falling up. Just because you've hit a bottom that seems like a negative spot, pretty much there's the only way you could go is up, and there's always a way to turn it into something positive. And I think in general, successful people are always good at failing. You have to be able to turn a failure into a positive somehow and not get frustrated or upset, and just learn how to be resilient and keep persisting and push through. So wouldn't say that's a particular piece of advice that anyone told me, but that's just something that I've been modeled in my own family, in my own culture, and all the scientists that I know, that you just have to figure out how to turn a failure into a positive experience and to keep moving forward, because things can always turn around.
Definitely. Well, Sydney, I think that is a tremendously important lesson. Thank you so much for sharing it. And is there any other last piece of advice or last note of inspiration that you would like to leave everybody with at the end of our conversation today?
Something I always like to impress upon people is that fungi are a really important part of your life, whether you know it or not. If you drink beer, if you eat bread, those are fungal products. If you eat cheese, that's also a fungal product. All the plants around you are associated with mycorrhizal fungi. They helped plants colonize land. Fungi are important for medicine. For example, penicillin is also a fungal product. So just understanding that fungi are really important aspects of your everyday life, even if you had no idea about that.
I love that. Listeners, we must have a greater appreciation of all of the fungi around us. I think that's a wonderful note to end on. Sydney, if our listeners want to learn more about you and the work that you do, what is the best way for them to do so?
I have a website, Sydney Glassman. I think it's at wordpress.com. If you just Google me, Sydney Glassman, UCR, or Sydney Glassman should show up my website. So my website has all of my research projects, all of my current personnel, my contact information, and all of my publications are there. So yeah, if you want to see all my research and all my papers, they're all on my website. Google Scholar, you could also search my name and find all of my papers as well.
Fantastic. Well, listeners, definitely take some time to browse Sydney's website, learn more about the fantastic work that she's doing. And Sydney, it's been such a pleasure to chat with you on the show today. Thank you so much for spending time with us.
Thank you for having me.
It was a joy to speak with you, listeners. Always wonderful to have you here as well. We hope you'll join us for our next episode of People Behind the Science.